Welcome, biologists, to this first of two screencasts that are going to cover the material for Chapter 7D, which is talking about the third line of defence. So that's our adaptive immune response. So in this first part, I'm going to talk to you about humoral responses and what they are and what that actually means. And then in the second part, I'll focus on the cell-mediated responses. So that's why I've broken it into two, also to try and keep them a bit shorter, quicker and easier to watch. But getting into it, our learning intention for this first part is to learn about the initiation of the humoral component of the adaptive immune response. And by the end, you should be able to explain how the adaptive immune response is triggered initially and the key role of antibodies in our defenses. So what is an antibody? What's the structure of an antibody? And how does it actually play a part in defending us against infection or disease? The study design, what we've already covered so far are some of the barriers. So that's your first line of defense. And then we've covered your second line of defense, the innate immune response, which is that second dot point there. Then you've got this third dot point, which is how do we initiate the immune response, antigens and how they're presented. We've talked already about the distinction between self and non-self antigens, but now what we're going to talk about is a little bit about what happens when you receive contact with one of those antigens. So that's our main focus, is the bit in red there. The third line of defense is just a reminder of the basic breakdown of our immune response. So you've got your immune system always scanning, always looking for pathogens or potential threats. The first line of defense are your barriers, innate and non-specific. The second line of defense, which are things like your phagocytosis, your complement system, they're all second line of defenses, but still non-specific. And then now we're going to get into an adaptive response and a specific response. It is important to note the key distinguishing feature there, which is it is specific as opposed to non-specific. In other words, each pathogen triggers a highly specific and targeted response. As a result, it's also slower, which is another key thing to remember, but we'll work our way through that as we have a look at the system. So what are the features? I've already touched on this just then, but what are the features of adaptive immunity? One of the key ones, again, is that it's specific. So the response is tailored to each individual pathogen. The response for an E. coli infection is not going to be the same as the response for a COVID infection, for example. And the other key feature is that it has immunological memory. So memory cells are a type of cell created specifically so that if you re-encounter that pathogen again, you generate a very rapid response. And we'll talk about that a bit more in the cell-mediated section in part 2, what memory is and what that rapid response looks like and how that actually works. So starting with one of the key points that your study design talks about, and we did touch on this when we looked at phagocytosis and the different types of phagocytes. We looked at things like neutrophils, macrophages, and dendritic cells. And we talked briefly about antigen presentation. So that's what this slide's about. This is actually from that presentation that I did on the innate responses in Chapter 7C. But here you've got that breakdown of antigen presentation. So it's basically phagocytosis. You've got your pathogen, which is the little red bacteria here. It gets eaten up by the phagocyte. It gets enclosed in a vacuole, fused with a lysosome. There's digestive enzymes in the lysosome which break down the pathogen. As you can see here, it ultimately gets broken down. And then the key difference between this phagocyte and some others is that it presents little fragments of the antigen. So not the whole bacteria, it doesn't take the whole thing. It breaks it down and then presents parts of the antigen on its surface together with an MHC2 marker. So if you remember, those MHC2 markers are self-antigens. And what these phagocytes do, these antigen-presenting cells, they're a very special type of phagocyte. And very, very essential to your immune response, as you're about to see. They take it and they hold it up like a beacon saying, here's this thing that I found. You need to come and do something about that. And it's literally just all it's doing is presenting it to other parts of the immune system to do their job. So T helper cells are an essential cell type in your adaptive immune response. They're essential in responding to an antigen. In order to be activated, they must see the antigen. They cannot see it unless it's presented to them. So the antigen presenting cells are the cells responsible for making that presentation, showing the T helper cells, here it is, come do something about it. And as I've already said, they present that antigen on their surface together with MHC2 markers. This is on page 390 of your text, showing you what I'm talking about there. Here is your antigen presenting cell. Here is the foreign antigen being presented together with MHC2. T helper cells will have specific T cell receptors on their surface that bind to that antigen fragment MHC complex. So it's not just the antigen fragment. They cannot recognize the antigen on its own. It needs to be presented to them together with the MHC2. And when they see that, that will then activate the T helper cell. The T helper cell has a role in both humoral immunity, which we're going to look at in just a moment, and cell-mediated immunity, which I'll look at in part 2 of chapter 7D with you. So these T helper cells, they have receptors that interact with the MHC2 antigen complex. They are unique, though, for a single antigen. So in other words, only one type of T cell receptor will bind to that antigen. In other words, only one type of T helper cell will bind to that antigen. Again, the antigen, therefore, will only activate that one particular T helper cell, which is what gives this system its specificity. Why is it called a helper cell? It doesn't actually do anything itself to destroy the pathogen, but it helps other parts of the immune system do their job, both the humoral system and the cell-mediated system. And we're going to look at those in a little bit. So I've touched on the fact that it can help with humoral and cell-mediated, but what's the difference between the two? So this short video, I'm going to play that for you in just a second, gives you a summary of what the humoral system is and what the cell-mediated system is. In broad terms, humoral, that we're talking about in this section of chapter 7D, is a chemically based response. And cell-mediated, as the name suggests, is a cell-based response. But I'll let the video do the talking for me, so I'll play that for you now.
Welcome to the video on humoral versus cell-mediated immunity. The humoral immune response is all about the B cells and antibodies. Its main function is to provide immediate protection against many bacteria and viruses. Since antibodies are produced from plasma cells, this is why this type of immunity gets its name. The Greek word humor refers to body fluid. Once a pathogen enters the body, then either the primary or the secondary immune response would happen, as we reviewed in the previous video. I want to elaborate a bit further on that topic, though, in regard to the antigen-antibody binding. So when a large molecule enters the body, it usually comes with many different antigens. Antibodies recognize very specific areas on that molecule, which are called epitopes, or antigenic determinants. The matching area on the antibody is called the paratope, or antigen binding site. Examples of humoral immunity include anaphylactic shock, eczema, transfusion reactions, and bacterial viral infections. A cell-mediated response happens after a T cell recognizes a specific antigen. It's also called delayed hypersensitivity. The main immune cells involved are, of course, the T cells, macrophages, and NK cells. Like the humoral response, the cellular response protects against bacteria and viruses, and also protects against tumor cells and fungi. Examples of conditions evoking a cell-mediated immune response are toenail fungus, tuberculosis, contact dermatitis, and transplant rejection.
So humoral immunity means a chemical response. In other words, cells, not just cells eating or killing things, but it's a chemical response. And in the adaptive immune system, we're talking here about antibodies. And antibodies are a protein, and we'll talk about the structure of them in a moment, but they're a protein that aim to neutralize or assist in the destruction of pathogens to protect you against both infection and disease. And there is a difference between the two. You can be infected with an organism, you might carry it, but then you eliminate it before it does any damage. So infected, but no real disease. Disease, by definition, needs to have some sort of symptoms. So that's the distinction between the two. When I say infection and disease, they're not necessarily, it's not necessary that one will always follow the other. All right, so we talked about what antibodies do. Let's look at how they're produced. So again, this is our humoral system. So there's a number of steps that need to happen. The cell responsible for producing antibodies is a B cell. All right, we'll get into some specifics about that in a minute, but it is a B cell or a B lymphocyte. B cells have specific receptors on their surface, and these receptors will again only be specific to the antigen of a particular pathogen. So one type of B cell will respond to an E. coli. One type of B cell will respond to a meningococcus, and that one's for you, Zach. All right, so the E. coli B cell cannot make antibodies for the meningococcus, and vice versa. It is highly specific. But in order for it to do its thing, it needs to be selected for. So the first step is selection of the B cell. How is it selected? Well, it will run around and randomly try and attach to various pathogens that might be present, their antigens. And eventually, it may encounter, only if you're infected, but it may encounter the antigen that it is specific for. If it does encounter it, then it is partially activated. All right, it binds to that antigen, but it can't do anything until it is fully activated by the T helper cell. So it binds to its antigen, and then the T helper cell, which is also separately come in contact with the antigen through an antigen presenting cell, it has now been activated. It finds its partner B cell and secretes a chemical called cytokines. These cytokines stimulate the B cell to both differentiate and start production of antibodies. All right, so the cytokines are secreted, they activate the B cell will differentiate. So it starts to divide through the process of mitosis, but it also differentiates, and it forms two different types of cell. One is called a plasma cell, and the other one is called a memory cell. The plasma cell is the one that produces the antibodies. The antibodies will have the same structure as these receptors that are on the original B cell. Why do they have the same structure? Because they are specific and they will bind to this antigen wherever they find it. So they secrete the antibodies, they'll travel around in the circulation looking for, finding, and mopping up these antigens. In other words, getting rid of the pathogen. The B memory cell, they just hang around in the circulation waiting to see if you ever encounter this pathogen again. And if you do, they very, very quickly differentiate and start pumping out antibodies as plasma cells all over again to clean up the infection before you really get sick. Right? That's the basis of the way vaccines work as well. You get vaccinated, you create these memory cells. So if you encounter the real infection, you very quickly develop antibodies. Hopefully, I'm going to ask you the question, then obviously because it's a screencast, I'm going to answer it, but hopefully you are starting to think about this already. How long does it take to get to this final stage here where the antibodies are in the circulation and killing the pathogen? The answer is about 2 weeks. 1 to 2 weeks minimum, right? So you get exposed to the pathogen right back here 2 weeks ago, and then finally 2 weeks later, you start producing the antibodies to actually kill it. Now, you don't want that to be the case if it's a really serious infection, because you might encounter the pathogen back here, and then by about here, you're dead. That's not ideal. All right, so if you get vaccinated against something, though, you've got these guys floating around, you encounter the pathogen, then very, very quickly, you don't need to go through all these steps again. Here's your memory cells. They go, oh, yep, I know what to do. And you skip straight to this step really, really fast. And massive amounts of antibody are produced. So you mop up the infection before it can cause you any real damage. Hopefully that all makes sense. We will go through this in class. And I appreciate that this is it's a 4-stage process, which in theory makes it simple. I know it's not. I know it's really tricky. So we will go through and make sure you understand the details behind that. So types of B cells, which you've already touched on, you've got your B memory cell. They have these specific receptors that are specific to an antigen for a given pathogen on their surface, and they just float around and won't do anything really unless they re-encounter that antigen a second time. And then you've got your plasma cells. They are the response. They secrete the antibodies because, hey, there is a pathogen here, better make some antibodies. They do, and again, that goal there is to clean up the pathogen. So we touched on selection of B cells, right? And then you'll end up with this differentiation, and then ultimately plasma cells with antibody production and memory cells. But how do we get to that point? The reality is B cells are called B cells because they live in your bone marrow, and they will be exposed to a range of different antigens. But you'll have a range of different clones, all right? So you'll have all these different B cell clones. The receptors on the B cells are all different shapes, And those receptors will only be specific to one particular antigen. All right, so up here you've got all these different clones, clone A, B, Z, etc. Right, and then they'll just float around waiting to encounter an antigen. And this antigen here, this little round antigen, well, it's going to bounce off A, it's going to bounce off B. It's not going to do anything. It's not going to select for the clone. But antigen X encounters B cell clone X, then B cell clone X is selected. All right, so that is the process of clonal selection. In other words, you've got all these B cell clones just sitting there waiting to randomly meet their antigen match. All right, it's like a dating game. They're just sitting there very lonely, very alone on a Saturday night, I don't know, watching bad television and eating ice cream. And they're waiting for their antigen to walk through the door and sweep them off their feet. And if they do, they're selected for. If they're selected, they then undergo a process called clonal expansion. So in other words, there's not many of these, just enough so that they can be selected. But now they've found their perfect match. It's like, righto, well, I need to spruce myself up, and they expand. What they actually do is, in other words, divide rapidly. So they'll divide through mitosis, divide and differentiate, creating plasma cells and creating memory cells. And again, your plasma cells that have been rapidly produced in response to this initial exposure, they make your antibodies. But do remember, there is that extra step. These B cell clones will, sure, they'll differentiate and they'll divide, but they can't do anything at all unless they encounter their T helper cell. Right? They need to encounter their T helper cell as well to be activated to actually start pumping out the antibodies. So there is an extra step that's not in here. That is an important double check, and I'll talk about that with you when we talk about autoimmunity. So I won't go into that now. It's confusing enough at the moment. But why do you need T helper cells? Why couldn't we just skip that step and start pumping out antibodies? And part of the reason is, what if your B cell clone is responsible for, or if its receptors actually react to your tissue? They react to your MHC markers. Then you start attacking your heart tissue. They start pumping out antibodies that destroy your heart muscle. That's probably bad, right? So there's like this double activation step where, yep, your B cell clone might recognize your heart tissue, but it still can't do anything unless the T helper cell is also activated. How is it activated? Through antigen presentation. So it's like a check and balance, right? That's more detailed than I should have gone into. Hopefully that makes sense though, and doesn't make this even more confusing. But we'll talk about that more when we talk about autoimmunity. So this is actually the image from your textbook. That last one wasn't. That's from a textbook I've used previously, but I think it illustrates it quite well, and in some ways a little bit better than this. Because this one, it's telling you the same thing though, right? Clonal selection, antigen X matches these receptors. So these ones are selected for. When they're selected for, they expand. And then it gives you that extra step, which is some differentiation into plasma cells and memory cells. It's the same principle that we just talked about, but different image. And it's the one from your textbook. This image is different to the one that's in your textbook. I have got the one from your textbook up next. But again, I like this one a little bit better. I think it explains the structure. You are required to understand the basic structure of an antibody. Okay, so what is an antibody? It has 4 polypeptide or 4 protein chains. 2 light chains and 2 heavy chains. All right, so here, these little short ones, they're your light chains, and this big purple one here is your heavy chain. All right, and each of these 4, because there's 2 on this side and 2 on this side, but each of these 4 antibody polypeptides have 2 regions. There's a constant region, so that's the main part of the antibody. So here it is actually the yellow part, and on this one it's the purple part. They're constant, they don't change. All right, every antibody has those. The key bit that makes your adaptive immune response specific is these little black bits on the end. The black bits on the end are what we call the variable region. So these vary in terms of their structure and in particular their shape. So different antibodies will really only be different in this little tip region in the variable region. The shape varies so that different antigens can fit that antibody. In other words, only one antigen will fit in that variable region. There'll be a different antibody with a different variable region that fits this circle. All right, or one that fits this oval, or one that fits whatever that is. Is that a pentagon? Yep, pentagon. All right, so one specific variable region on an antibody will fit one specific antigen, and the variable region of the antibodies is what creates the specificity, and that's a really important concept. In terms of what you need to know there are 2 light chains, 2 heavy chains, and each of the 4 chains are divided into 2 sections, constant and variable. All right, that's really the main level of detail you need to know. The only thing that your textbook illustrates, which is important, that previous image did not, is the binding between the two chains. So it's a disulfide bond that links the light chain to your heavy chain. But everything else that you're seeing there is the same. There are different types of antibodies. All right, so the main ones that we talk about, and really they are the only ones that you need to know, and you don't need to know these in a lot of detail. What you do need to know is there are different antibodies that have different functions, and they have different features and characteristics. So IgA, which is found in like your tears, your saliva, mucus, breast milk. IgD, which really just activates immune cells. IgE is particularly responsive in one of the things we look at in particular is allergic reactions, but also if you have a parasitic infection. IgG is the most common antibody. I'm going to talk about that again in a second. And IgM is the first antibody produced. All right, and that's really important. Each of these are produced at different stages. Your first response is something called an IgM antibody. And the IgM antibody gets pumped out, and it's actually the first sign that you have an infection. We can actually test for IgM in your blood for a specific virus or bacteria. And if you've got IgM, it means that it's a current infection because your body pumps that out first. It is currently responding to that infection. As that response matures, it stops making IgM, and then IgG takes over. IgG is the most common antibody. It is the main responding antibody to infections. And it is a small antibody, and which is important because it's actually small enough to cross the placenta. It helps protect babies before their immune system develops because it can cross from the mother to the baby and protect them. In some cases, it can also lead to the mother's antibodies attacking the baby, which can be an issue as well. But we'll talk about that later as well. So what do antibodies actually do? There's 4 key things they do. Again, this is from your textbook, and I have another image that will help explain this a bit more. But neutralization, the antibodies can bind directly to the surface of bacteria, toxins, or viruses. And what they do is they inactivate them, neutralize them. The bacteria needs these sites to bind to your body or to invade. If you block them, it actually can't invade. This is the principle behind the COVID vaccines, right? It targets the spike proteins on the surface of the coronavirus. So that's sort of what this image is showing here. These antibodies are stuck. This is not coronavirus. It's just some picture. All right, but let's pretend it is. These antibodies stick to the spike proteins on the surface of the virus. The virus relies on the spike proteins to invade and get into a cell. If you block them with antibodies, it can't. Right? That's the whole concept behind the way the vaccine works. It targets the spike proteins, neutralizing the virus, because if the virus cannot invade a cell, it cannot reproduce, and therefore you don't get sick. So that's the whole concept there. Agglutination, this is showing you with red blood cells, but it can do the same with bacteria. It will bind to antigens on the surface of the cell and cross over between multiple cell types, binding them together. If this is bacteria, it actually means that they can't invade, they can't attach where they need to, they can't feed properly, they can't multiply properly, so it kills them. All right? And so that's a couple of different ways that they can help protect you. We talked about opsonization when we talked about phagocytosis, but if you stick antibodies all this over the surface of a bacteria, and they have these little red beacons on the end of them, like a flag, and that flag basically is like waving a signal to the phagocyte, hey, come eat this. This is not meant to be here. Come and eat it. So opsonization is coating the pathogen with antibodies to send a signal to phagocytes to come and ingest the pathogen and destroy it. And then we talked about in the innate immune system, your complement system. So antibodies can help trigger complement activity. And so then when we look down here, this is the same sort of summary, but with a little bit more detail. Neutralization, blocking the surface, agglutination, sticking them together, precipitation of dissolved antigens. So that's sort of agglutination as well. We didn't talk about that one before. All right, and then all of those can help enhance phagocytosis. And then over here, activating complement, and remember, complement leads to cell lysis through the membrane attack complex. All right, so that's our humoral immune system. There's a lot to take in there, and immunity is really complex. So we're going to unpack this. We're going to review this. We're going to do questions around this. But I'm hopeful that this helps with some reading you've done over the holidays, hopefully the work you've done around your summary in preparation for coming back to school. And what I encourage you very, very strongly to do, make notes, but particularly notes about your questions that you've got. Ask me stuff. Tell me what didn't make sense. Preferably don't just say all of it. If that's true, then I guess that's true. But target your questions. Let's really unpack this, because yes, it's complex, but it is possible to learn it. So we have to work together on that, and we need to keep moving through. So come with questions, come armed, ready to go. I look forward to seeing how you went with it. And yeah, we'll unpack this together when we're back at school on site next week. Thanks very much for watching.